It all started with this microbe that was found on the border of a dry lake bed in a spring. You see the little dots inside that microbes are sulfur granules. And it turned out that this microbe is a sulfur oxidizer. And that was quite interesting because it lived in a spring on the side of a dry lake bed. And that lake is wet in the spring after winter when you have ice falling and lots of snow melting. In the spring you have some water in the lake, but on the side of the lake you have two springs that are mesothermal. So room temperature, like our swimming pool temperature. And you have those microbes living there. They are white. They are sulfur oxidizers. They are aphotic, so they do not need sunlight to survive. And actually, they do not like oxygen that much. They can tolerate it in small concentrations, but they would rather be in areas with small concentration of dissolved oxygen. And the thing that's very interesting is that this is an open air area. There's sunlight going there. So that's why it was interesting to see those microbes there. And where they actually come from, where we actually find them more often is in caves, underground, in little forts, or just water flowing underground. And yeah. Here, the geologic setting of the area, it's in Oregon, and it's a volcanic area. You have volcanoes are pretty far away, but it's still a volcanic region. There is that huge valley with lots of lakes, and the water is running off from those springs, from those hydrothermal springs, towards the center of the valley. And this is where I took samples. So this is what the dry lake bed looked like. You have vegetation on the side of the lake bed. You have the springs on the side. And you have two springs next to each other. This one is the spring where we found the microbe. And this one, we did not find any microbes, but that spring goes on the ground most of the time. So it doesn't mean that the microbes are not there, it's just we don't see them. And so the point was to take samples, take the core sample. So like you could see here, it's about 1 meter tall, but when you actually get the sample, it's 0.5 meter long. And so we took samples along the flow path of that spring and along the one that is just a few hundred meters away from each other. We looked at the sulfur compounds in those samples to look, and we look at the isotopic values as well of those samples. And the point is to find if there is something interesting happening at the location where those microbes are actually thriving, even though it's not their optimal living condition, living area. They're still there, and they're still very happy to be there. And so we wanted to know if there was something interesting about that area. And the short message is it is interesting. The problem is that all of those areas are all different from each other. So even along one spring, each point of the flow path is different. And so here, like take a deep breath, but we have sulfur concentrations, and so that's sulfur concentration. This one is exactly the same, but in a different view. Here, the sulfur concentrations are based on the amount of sample I had, so it's the true concentration. Here, it's a concentration relative only to the sulfur compound. So 100% is 100% of sulfur. So overall, if you take all those points and you add them at one depth, you would get 100%. It's the sum, 100% is the sum of sulfide element of sulfur and sulfates. And it's just a different view to see if we could see something, because most of the data we got is just all those ziggly lines going everywhere, and it's kind of hard to see anything happening. Then we looked at the sulfur isotopes, just to see if there was something interesting. Usually you have fractionation, so having the microbes preferring light isotopes. Usually you have that for sulfate reduction, and you don't see that much for sulfur oxidation. So we are looking if there is sulfate reduction or anything. We did assume there would be sulfate, and there would be sulfate reduction, but actually there was really, really not a lot of sulfate in any of the samples. And so if you look at the true concentrations, so here you have so the spring that doesn't have a lot, that doesn't have the microbes, and here is the spring, another, the second area, and then the dry lake bed. Those concentrations are very low. Compared to marine settings, I mean, marine settings are sometimes percent, or at least per mil, not ppm. So there is a 100 or 1,000 difference in order of magnitude. So that was actually interesting that the dry lake bed, so on the photo where you see me take the sample, and it's all very, very light colored and very bright, there was absolutely nothing. I mean, it's just very, and so that's kind of a little bit depressing, because it's actually where that would be the end zone for preservation potential, and it actually doesn't preserve anything much, which shows that, if you go on Mars, and if you did have a cave of some place on the side of maybe that big valley on Mars, and that you'd have some seep coming and bringing with it those microbes that live underground in that cave, in that lava tube, whatever you want, and bring them there, your rover really has to be at the right spot to see, I mean, at least those microbes. And so, because a few hundred meters away you might miss it and not really see it. This one here is the one where we actually see the microbes, and it's just, everything is a little bit different. Here we have a lot of sulfates that we don't have much here. It's going to take a lot more thinking about why this is and what exactly is going on everywhere. But it's actually very interesting. Just one night, those are, I mean, fresh results to see that all those isotopic profiles and even the concentration profiles are all different, even so close from each other in area. One thing that is interesting is the width of the isotopic range. It's not that big like for marine settings, but it's still pretty wide. And there is definitely something different between the spring that actually has the microbes and the other one that's next to it. This one is much broader, whereas, this one has a narrower range of values. So we'll have to do some statistics on that. One thing here is actually the fractionation pattern. Not that interesting right now. I'll have to you know, I'll have to study that a lot more. But something that is actually visible is that there is here, we do see a pattern with the fractionation of the so it's sulfate compared to sulfide, or elemental sulfur compared to sulfide. And so that would be looking at the sulfate reduction, whereas this one would look at the sulfur oxidation. And so there is definitely a pattern having all that there is fractionation, the fractionation is kind of different between the two. There's something I forgot to mention here on the big profiles is that I changed the color here of some of the data points to show which of the species, of the sulfur species, is dominating the actual isotopic value. So here, as I said, those ones are not the raw data about the sulfur isotopes. Sulfur isotopes would look like something like that. So here it's actually the concentration multiplied by the isotopic value, and that gives you a whole average for that depth. And so you can see that at some depth you have the elemental sulfur that is actually dominating the isotopic value. And here on the Foscut Lake, you have sulfate dominating the whole isotopic value. But most of the time it's the disulfides. But always, to keep in mind, it's always small concentrations, never something in the order of magnitude of the ocean. So that's what I'm going to present today.
Thank you very much. Do we have any questions for Cindy? All right.
So I was wondering, you said that there's some that grow like out in the open, exposed to sunlight and air, and then some that grow in the cave. And they're probably the same genus. Has anybody done any like species or subspecies genomic information on them to see what the differences might actually be and why some of them will grow in the presence of sunlight and on the lake beds, but then others stay in the cave?
So they have cultured the one in the cave. Those ones, we haven't tried to culture them at all. We haven't even looked at anything. We just, looked at some of the DNA and matched to see what they were the closest relative to. Why? I think it's just a tolerance thing. If you've got enough food and you don't care about the sunlight, you can be there. They can do it very well. And the one thing is there's a lot of grass in the summer. So at least for the oxygen, it's possible that the decomposition of that grass, even during the summer or in the winter, will decrease the amount of oxygen in the water and will at least make them in a zone that is almost anoxic.
Cool. Anyone else? If not, we are now, thank you very much. Well, thank you again. And then we have a little break.